All right, if I could have your attention, please. So thanks, everybody, for coming out. I have Mike's attention, at least. I'll be introducing the speaker today. I'd like to introduce you all to Rosy Joshi-Mukherjee. Rosy has, as you'll hear when I diagram her CV, has literally worked at some of the best labs in the cardiovascular field in the basic science area. In 2008, she got her Ph.D. at SUNY Upstate Medical University with Mario Delmar as her advisor. After that, she went on to do 2 postdoctoral fellowships at Johns Hopkins University with both Gordon Tomaselli and Les Tung, who was the department head, I think, also at that time for biomedical engineering there. Joshi has had a very good smell for like what's going on, what's really hot in the biomedical research field, starting off with her early identification of the connexin 43 C-terminal fragment, and then some really fascinating work with something called arrhythmogenic right ventricular cardiomyopathy, where she characterized 2 different mutations leading to ARVC. And then now her current work, which is really exciting, I think it's what led her initially to a small consortium, Aurora Research Institute, which is really looking at these hearts on a chip. How do you use a human-induced pluripotent stem cell model to actually predict patient responses? And I think Joshi's going to talk a little bit about that. And so she just received, I think, an FDA grant on some of the work she's going to present. And I'm not going to spend too much time because I'm very excited, I think, as everybody else is, to see what you have to present. So thank you, Joshi.
Thank you, Dr. Posing, for introducing me, and thank you to all the committee members who gave me the opportunity to come and share my research, and thank you all for coming. I hope you don't sleep. This is late in the afternoon, so I'll keep you awake, hopefully. Today my talk, I've divided my talk into 3 different sections. In the first section, I'll talk about what I've done or what my research is about, in which I'll introduce you about the induced pluripotent stem cell technology. And in the second section, I don't know why the movie is not going, but maybe. Okay. Oh, yeah, that's true. I was thinking something went wrong with the video or something. So, and then so why we are interested in making these induced pluripotent stem cell is because my focus is more towards the cardiovascular research. So how this technology we can use to our advantage to produce these cardiomyocytes. And we all appreciate how beautiful these beat like a human heart beats. And then in the second section, I would use, I'll introduce you to a multi-electrode array technology to measure the electrical properties of these tissue. And not just the electrical properties, but how we can measure their action potential in a very unique way than generally is being used by either optical mapping or patch clamping. But this is an alternate way of how we can measure action potentials. So, and then in the third section, I would tell you where my research is headed and what I can, if given an opportunity, what will I bring to Virginia Tech. So with that, so we all know how the heart disease is the major problem in the United States. You can see very well here how red this map is. And 6 million deaths are happening due to heart diseases, 1 in every 4 deaths, which is alarming. And 5 million of the cases are reported to have congestive heart failure. So heart diseases are concerned, and the next 2 decades, they are going to increase. And the cost associated with this is also going to increase. So we need to, address it.
Can I ask you a quick question about
Yeah.
that graph? So I've seen that graph before, and my eyes are drawn to Minnesota. I think that's Minnesota.
Yeah, there's something
there.
there.
White. So what are they doing right? Is that genetics or something in the lifestyle or what?
I think it's the lifestyle. Maybe they eat a lot of cheese or their food and the lifestyle relaxed Midwest. We should all go to Midwest. Yeah. So, so there are several drugs that are in the market which treat cardiac diseases, but over the period of time, we have also learned that they don't translate very well. And 35 of the drugs which were approved by the FDA were recalled. And these are some of the examples of the drugs that I find interesting, that they were not cardiac drugs, but these drugs were treating some other problems. But you can see in most of them, they all led to death associated with cardiac. So they were recalled. And even though they were in the market for so many years, we did not understand what was going on with these drugs. So both cardiac drugs and non-cardiac drugs can also lead to cardiac-associated problems. So so going back, what are we doing wrong? I mean, there are animal models. There are the molecular mechanism of a lot of these heart diseases are being scrutinized too, so that we can understand. But I think one of the things maybe which we are missing is how we are translating that into the clinic. And we all know that the physiology of animals are much different from the physiology of the human. So is that the main problem? Or is it something, there's a missing link between from what we do in the lab to what we actually, when we treat these patients, what is there more complexity involved that we are missing? So the missing link was recently being proposed to be addressed by another alternate model system. And we are not saying that we should shut down all the animal research and just move to these new model system. What we are saying is we can provide an alternate way of looking at some of these problems, which could give us more insight into what the effect of these diseases or the drugs are happening at the molecular level. So this alternate technology is the induced pluripotent stem cell. So what is unique about this? What is the fun part about this technology? So this was pioneer work done by a scientist in Japan who also got a Nobel Prize for this. What he did was he took a cell type from adult human patient, and then he put some transcription factors, as you can see here. Oops, sorry. Transcription factors as he narrowed down from 24 to 4 factors. And when he put them in the skin cells, he converted magically these mature cells into stem cell-like phenotype. And this whole pathway revolutionized the way we see medicine now. And the other aspect of this research is, I didn't ask, do people can hear me nice and clear? Another aspect of this research, of this technology is human, right? So before that, we were mostly focusing our research on cell lines that either were, they were not patient-specific. So these are derived from a specific patient. So these are your cells in a dish. So your genome in a dish is very personalized. So if any person carries a disease, it retains in his mature cells. And when you put these factors in, that disease is persisted in these stem cells. So then you have generated a disease model in a dish. So the next, I would like to focus on what this, like the timeline for this whole process, reprogramming process. So here is the patient. He decides to donate tissue. It could be a skin tissue or a blood. More and more people are, blood. I've donated blood. I think it's easy to donate your blood. Less and less people won't want to donate their tissue as skin tissue. So preferred way is like peripheral blood, and it's now the protocol has been optimized to reprogram many different kind of somatic cells. So that's pretty standard. So you bring that to the lab, and then you put these I'm going to call them Yamanaka factors, and that's pretty standard in the field now. Everyone calls them Yamanaka factors. They are nothing but those 4 transcription factors. And so you put them in the Sendai virus. And why Sendai virus? Because it's non-integrating virus. And so you don't have to worry about being present in your cell line all the time, so it goes away. And then you put them, and then it takes about, this is a very long process, still not very optimized, so it's about, for the colony to appear, it takes about a month. And then once you get the colony, then you have to expand and characterize and confirm. All of that process takes time, and then once you have that step confirmed, then you can do all the fun stuff. So that's step 1. And the step 2 would be converting to your cell of interest. So if the disease is cardiac associated, you would want to see differentiation towards that lineage. If your interests are neuronal, then you can convert whatever cell type you want to of your interest are. So basically, for this reprogramming process, there are very complex signaling pathways that crosstalk and maintains their, once you have attained the pluripotency, you need to maintain the pluripotency. Otherwise, that model is no use because it's undifferentiated or differentiated state. You have to control that balance very well, and you want to control it for your interest, so you want timely differentiation towards a specific line. So this is for maintaining the pluripotency. You can see the main signaling pathways, PI3K, MTOR, who down the line activate pluripotency self-renewal related transcription factor. And there is a very delicate balance provided through the beta-catenin pathway. And if that balance is lost, this phenotype is lost pretty easily. So, and then once you want your pluripotency stem cells to differentiate into your specific cell type, here I'm focusing on cardiac lineage. Again, you have to maintain a delicate balance on these signaling pathway through Wnt signaling. Wnt signaling is very much associated with cardiac differentiation and through beta-catenin, and because it targets gene related to cardiogenesis. So going back to this initial transcription factor, as I mentioned, it's I would describe it a little more about how do you characterize these IPS cells are actually IPS cells. So you first expand these cell lines so that you can pick your colonies, and this is just an example under the microscope that you see a nice tight colonies that appears, and then you expand them, and over days they grow. And this is the picking of the colony is still very manual. It's labor intensive and very expensive. And it needs a lot of babysitting and taking care. The more you ignore them, they will differentiate into randomly in the culture, and you lose your cell line. So you have to be very careful. And the other thing too, you need to mark these with pluripotency transcription factor to show that they are maintained in a very pure form. So here, OCT4, NANOG, they are all pluripotent marker, and you can see they're expressing in the nucleus as in the merged picture here. And similarly, the SSCA4 is also a pluripotent marker which appears on the membrane. So there are other differentiation marker that pluripotency marker that you need to confirm by doing 3-germ testing, which I'm not showing the results, but that's another way of showing that you have a nicely reprogrammed colony. And also you have to show that there is no germline abnormalities in the chromosome so that your reprogramming method did not introduce any alterations. So once you have attained that reprogramming status for your patient line, then you would like to differentiate. So this is another process. So first you make the IPS line, and then now you want to differentiate them into cardiomyocytes. So here you can see that is again a timeline, but it's much shorter than the reprogramming itself. So here when you plate these IPS cells, you treat them with the Wnt signaling pathway, small molecule, and CHR is one of the negative. It's an inhibitor of GSK3 pathway, and so it activates Wnt signaling pathway and initiates the differentiation more towards the cardiogenic potential. And then you have to inhibit the Wnt signaling pathway through IWR, another molecule, to finally produce cardiomyocytes, which start beating, as you have seen earlier. That we have here I'm showing you a protocol that we optimized in the lab to produce a monolayer method of differentiation, which we were able to successfully differentiate these cells into high purity of about 90%, that they beat nicely in a monolayer. So again, when you differentiate the cells, you have to prove that they have nice cardiac marker. And you can see here the marker that we are staining is with alpha-actinin, which is cardiomyocyte marker, and connexin 43. And they form, actinin forms striated pattern, and connexin 43 is on the membrane, and some intracellular staining. Similarly, troponin I is a striated pattern, as you can see. So we can see that they are widely expressing these markers. So the question is what purity is our culture. So for that, we assay for these cardiac marker by using flow cytometry. And you can see that in our culture, monolayer culture method, we attain about 90-95% cardiomyocyte, which is pretty good in a standard in high percent cardiomyocyte cultures. So the next, we want to, functionally, we want to know whether these are a good cell model for studying cardiomyocyte electrophysiology. So you can see that they are spontaneously beating. So that is the characteristics of these cell model. And by patch clamp, we showed that they are, although I don't like calling them nodal atrial and ventricle, but in the field, this is what they are referred to as, is a mix of population of atrial, ventricle, and nodal-like phenotype based on the morphology of the action potential and some of the characteristics. But as you can appreciate here, even though a lot of paper that they are published, they produce these little action potential waveforms and start claiming that they are 3 different phenotypes. But you can, here in this graph, you can see that there's a huge heterogeneity in not only as a atrial in green, see how different it is, but also within the ventricle-like phenotype. And although we are saying that in our cultures, 75% of them are ventricle-like, I would rather call them as cardiomyocyte because there is a huge gray area for them to be claimed as a one specific cell type. Going back here now to in this, so far in this section of my talk, I just discussed about how IPS technology has been advanced to produce these cardiomyocytes in culture. So now we would like to move to my second section of my talk, where I want to show you how multi-electrode array system can be used to understand the electrophysiology of these IPS-derived cardiomyocytes. So what is this multi-electrode technology? So here, you can see any electrogenic cells when you put them on top of this chip. So this chip has a sensor which can then sense the movement of the ions in and out of the cell. And that exchange of ions can be then sensed by these chip to produce these extracellular signal called field potentials. These field potentials are the electrical signal usually people associate with this, as a surrogate to EKGs in the heart. And so this is pretty standard in the field to use these multi-array technology to assess their electrical activity. And the one of the advantage of using MEA is these are non-invasive, so you can study for a long period of time the behavior of these cells in the presence of the drug or in the presence, if there is a mutation associated with disease, then you can understand how that influences their electrical activity. So in the next part of my talk, I will discuss about this electrical activity of these cardiomyocyte, but in addition, I would also show how we were able to record action potential from using the same technology. So, so here is the beading monolayer on these MEA dish, just to show you that we could produce functional beating cardiomyocyte on these surfaces. And just to give you an overview, this is how the plate is set up. It is about 24 well, independent wells. So each well is a one construct. And here is the arrangement of the electrodes within one chip. So you plate, so what you're seeing here is one well, and the cardiomyocytes are cultured on this well. So at that well, there are multiple independent constructs that you can then measure. So here is just a representation of one well, how the electrical activity in each electrodes are represented based on what's sensing in these, from these cardiomyocytes. And here is just an expansion of one of the window. So what I'm showing here is we can measure the electrical activity of these cells. So then we went into addressing how we can measure these action potential. It's a very interesting story that in the lab we were just like, oh, maybe we should electroporate these cells and see what we find. Because, and then we were looking in the literature, and we know that electroporation is detrimental to the health of the cells. But what if you electroporate like you electroporate a micropatch? If the cells are very close to these chips, so can we gain an access to inside of the cell? And to our surprise, that's what we found. We gave a very small electrical impulse, like 1 volt, for about 1 millisecond and for about 30 seconds. And we at 1 hertz spacing. And you can see that prior to electroporation, they were like field potentials. But during the electropораtions, because there's so much perturbation, we did not see much activity. But as soon as we stopped the electroporation, we start seeing this phenotype that looked like action potential. And you can appreciate more in this window here when we extracted the post-electroporation, this window here. You can see that each of the waveform is now represented as a train of, like it's a train of action potential. When we superimpose them, they showed that they are pretty consistent from beat to beat. And not only that, we also show that we could electroporate them multiple times over days, and we could still get really nice shape of these action potentials. And similar action potentials were recorded using patch clamp. So they were very similar the waveform that we obtain from these multi-electrode array-mediated action potentials were comparable to those recorded from patch clamp. So that was amazing, because then we said, oh, that is a really nice way for us to understand how the cells are maturing over time. So because remember, these are just day 10 old cardiomyocyte that you differentiated, and we are hoping that they have those beautiful ion channels on the membrane to produce these action potential. But by patch clamp, we can only do one at a time, and we are limited by few cells, and that we cannot even follow them in time. So using this technology, it will be really nice to see whether we can see a consistent waveform, following multiple electroporation. So one of the hypotheses was, like, can we successfully record these action potential, and whether it's dependent on the amplitude of these field potential. The field potential amplitude depends on the maturation of the electrical part of the membrane based on the expression of these ion channels. So as you can see, as the cultures mature, the field potential amplitude goes up. So we were wondering if we could measure these action potential when the field potentials are really small, or if they were really big, whether that influences the shape of these action potential. And initially we thought it would, but to our surprise, we did not see. Actually, we could get really nice action potential at even lower field potential amplitude. So maybe we were thinking they were immature here. We were thinking they were more immature here. But I think that is not the only way to characterize, seeing that the maturation may not be actually translated into the amplitude of these field potential. This could be how tightly the cells are sitting on to the electrode, what the mechanism. At this point, we do not know the mechanism of how we are able to record these action potential based on just simple electroporation-based technique, but through the literature, we see that the electroporation may be causing a little nanopores on these membranes so that we could access the inside of the cells. So, however, here we can show that we still can measure action potential independent of the amplitude of the field potentials. Yeah?
So analogous experiments in the central nervous system, people record local field potentials, patch clamp single cell, and also a condition that's sometimes referred to in neuroscience as quasi-intracellular recording, or sometimes called IMC, imminent membrane contact. And in those situations, the way people view it, I think, is that you're kind of pushing on the membrane of the cell and shunting it out a bit, even though you have no patch on the cell.
Right.
Versus, at least in the brain, the local field potentials are very much related to the geometry of the cells in that area, and you're picking up a field from multiple cells at the same time. So I want to make sure I understand what you're calling a field potential here. That's still from an individual cardiomyocyte, not a field summation of longitudinal occurrence from many cardiomyocytes. Is that correct?
So you're talking about the field potential, this
yes.
one?
Yes.
So they are all from the network of cells, so they're not from individual cells.
Okay, so it is
So,
a true field from
yeah.
cells from the current flowing
Right.
in the extracellular space.
Yeah.
Okay. And then so the underlying action potentials from the individual cells, because they're so well
Oh, again,
synchronized.
these are not individual cells. They are from the same cell site. So if I go back here, so here we can, I'm showing you from one valve, one electrode, and they are field potential before the electroporation. So then when you electroporate, these signals get converted into more action potential like phenotype. We don't put any physical pressure or any perturbation related to.
So those action potentials are some action potentials of many cells simultaneously?
So yes, so here like you can see that their network of cardiomyocyte, they are sitting underneath these electrodes. So that's where, like when you electroporate, you can pick this electrode to electroporate the cells nearby. We can also electroporate this simultaneously, all of them.
And because they're synchronously firing,
Uh-huh.
it
It
very
appears
tightly
to.
looks like a single action.
Right,
That's
yeah,
awesome.
yeah,
All right, thank you.
yeah.
Can I ask specifically what's the window of time for when you go back to the field potential?
Oh, so it varies. That's a very good point. So it depends. So you're talking about here, right? So after this goes back to when does it go back to this point? So we have seen anywhere it's like highly variable. So sometimes it takes a day, sometimes it takes within a few minutes, sometimes it takes a few seconds, like 30 seconds. So it's very hard to know what controls it, but it's from electrode to electrode. We have seen different time points when they go back to the field potential. But at least to the point where we electroporated for 30 seconds, in majority of them, we see at least 15, 30 seconds. But there are places where next day we have come and still action potentials. So yeah, so again, I was here. So the question was whether we could simultaneously record from multiple cell sites over consecutive 2 days. So here we are showing day 1 recording from the same network, and then the day 2 from the same network. So you can see the example. These are just examples of action potential waveforms from these. So I think we all can appreciate how these are very minimal filtering. And this action potential shape of these from each of these electrodes are pretty robust and very nicely. These are the amplitude, we have normalized it so that the analysis is easier. The shape of the action potential, so the different color represents the each beat. So what we did was we superimposed from beat to produce the main action potential. So you can see that there is a little bit of variability, but not a huge variability. It's pretty consistent within the network. And also, the other question we had was whether we can, from multiple wells, like I showed you the way that it's arranged, whether we can record simultaneously from different electrodes, from different constructs, and so that it can make the system more robust and reliable. It's not just one waveform that we are showing. It's like we can produce it across multiple layers. So you can appreciate now, like from different constructs in day 1, they produce action potentials look like very similar to on the day 2 from the same network as shown here. So that brings us to the point, like, can we use this system to study drugs? Will the drug produce the same effect on these action potential that we have just recorded using this methodology? So one of the drug that we tried was with the beta adrenergic pathway. And the two drug that we tested was norepinephrine and isoproterenol. They both affect beta adrenergic pathway. And you can see in the case of field potential, the beating rate increases with time, with concentration. And also the field potential duration shortens with increase in the concentration. And it's nicely plotted here. You can see that with the increase in doses, the percent APT goes down. And similar results were obtained for ISO. So basically, these pathways are active in these cells. And also, we can use our approach to measure action potentials in the presence of these drugs. So now the question is whether we can produce some arrhythmic behavior, and can we use this methodology to identify or to record that. So for that, we used E4031, which is the IKR blocker. And you can see appreciated here in the field potential, it gets the RR interval increases because you're blocking the IKR channel. And similarly, in the case of action potential, they get prolonged over increase in dose of this E4031 blocker. And in the case of 100 nanomolar and above, you can also see there is an appearance of arrhythmic behavior in these action potential as produces arrhythmogenic EADs, so which are associated IKR blocker properties. Yes? For some reason, I can't explain that. I think the reaches a threshold. I don't know. No, it goes really high up here, or maybe the way we are measuring APD 90, like in these arrhythmic traces, to tell you the truth, I don't know. Yeah. But it's longer than the, at lower concentration, but this is the highest we saw, and then I don't know why it goes. We didn't patch clamp, or we didn't do any of those, so. That's a good point, but so the next, I mean, so far it's all physiological. We block the channel, we are prolonging the action potential. We activate beta-adrenergic, and we increase the beating rate. So the next question is whether this model can be used to study some of the cardiotoxic effects that people report in the literature. And for that, we used a doxorubicin. Why doxorubicin? Because one, we know that this is a good system to study because it causes changes in the altered rhythm. So doxorubicin is an anthracycline, and it's used for treating cancer. And in cancer patients, people have noticed that over a period of time, they also have cardiac-related arrhythmia and heart rhythm alterations. And also, it also causes cardiotoxicity, means there's a lot of cell death. So here, I'm showing you a dose and time-dependent effect of doxorubicin. And you can see as you goes over, as you increase the dose, there is a significant cell death above 1 micromolar concentration. But at lower concentration, at 0.1 micromolar, there is not much significant effect on the phenotype of these cells. So there's not much cytotoxic effect. So for us, the most interesting thing was in this dose range and in this time period, where there is some cardiotoxicity here, but there's minimal cardiotoxicity, so to know what is happening at the electrical level in these cardiomyocytes. So we measured using just field potential, and you can see the top is at 0 hour, and as you see over this, like over a longer period of time. So initially, all of them, there is no alteration in their beating rate. Even after 1 hour, putting the drug on these valves did not cause an effect. However, when you go back to 24 hours and longer, here I'm showing the green represents the 1 micromolar concentration, and the blue represents the 0.1 micromolar concentration. So the 0.1 micromolar, you can start seeing there is a slowing down of the beating rate, but they are still consistent, whereas the 1 micromolar is all over the place. So they are already beating irregular. As you increase and go further in time, majority of the 1 micromolar, the electrode that constructs were not active. They did not, or they were not showing any beating, so we couldn't measure anything. Whereas In the lower concentration, as you remember, there's no much cell death whatsoever in higher, with increase in time. So, but however, we can see an altered electrical pattern across days. So something is going on, and using doxorubicin, we can tease out how doxorubicin at a very low non-toxic dose causes these electrical disbalances in these cells. So it's a very new data that we got. We still are investigating this field. I'm not going to say much more than this, but what I'm showing here is we can, using this sensitive technology, we can study drug-associated alterations in the electrical behavior of these control cells, which, I mean, is a nice model system to study any cardiac and non-cardiac drug. So now I have shown you all my first and the second part of my talk. Moving on to what can I bring to Virginia Tech, or where my research is heading. So going back to my signaling slide, my favorite slide, talking about what is going on in the pluripotency and in the case of iPSC differentiation. So as I mentioned already, that there's a delicate balance of these two pathways, the PI3K, AKT, mTOR pathway, and Wnt signaling pathway. It's been studied in the case of reprogramming. But what I also did not, I mean, what I should mention here is we do not have a good understanding of the reprogramming mechanism, because even though the Yamanaka factor has been identified, the efficiency of reprogramming is only, in a good transduction, about 1%. So we still do not know the underlying mechanism of how these pluripotency self-renewal status is attained. So, and the other thing, like when you want to differentiate, we have much better handle in this pathway for producing high percent cardiomyocyte, but we still do not understand to the level that we can produce a very pure population of mature ventricular atrial or nodal cells. So that pathway is still not well characterized. So what is also recently been shown in the case of connexin is the gap junctions are critical for reprogramming somatic cells, because when a recent research finding shows that when they express these connexin 43 during reprogramming along with the Yamanaka factor, they increase their reprogramming efficiency significantly. And also when you also the connexin 43 plays a big role during the differentiation process. However, what we do not know is how this pathway is regulating the connexin 43 and the gap junction. That mechanism is not well studied, and we do not know much about these two, how they connect, how connexin 43 is regulated during reprogramming. That is what my interest is. So that's what leads to my future research, is how does a cell regulate connexins during somatic cell reprogramming, pluripotency, self-renewal, and differentiation. And my main hypothesis is Yamanaka factors, like when you introduce them in these somatic cells, will regulate connexin 43 and its fragment, gap junction 20 kilodalton isoform during these reprogramming process and differentiation. And this hypothesis also stems from my Ph.D. research, and also in recent year from Robin's lab, Dr. Robin Shaw's lab, and also Dr. James Smith, who very well characterized how these 20 kilodalton fragment of connexin 43s is produced. So just to give you a brief overview of people who do not know, so connexin 43 is a 43 kilodalton protein, and it has 4 transmembrane domain and an N terminus and a C terminus, and 2 extracellular loop. What interesting was the way when I was working for my PhD in Delmar lab, we unknowingly put a stop codon in the middle of the gene, and we did not know. And then we put that vector in the cells, they started glowing. And we did not know why they started glowing, because we figured out later on that by sequencing these, they have a stop codon. And then when we ran a Western for that, we found these fragments of connexin 43, which should not have been produced if there was a stop here. So later on, from Robin Shaw's lab, it was really nicely shown that they have many internal start sites that could be regulated to produce these fragments. And unfortunately, that also was a part of my thesis chapter for my PhD, which never got published. But at least I know that the field is like confirming that these fragments exist. And so the transcription regulation of these 5' of connexin 43 leads to these different fragments of protein in the cell or machinery. And that role of connexin 43, 20 kilodalton could also play a role in reprogramming and differentiation. Is that what I have a strong interest in? So my main objective is to understand the mechanism of these internal translation initiation of connexin 43 and how that internal translation will modulate these 20 kilodalton isoform in reprogramming and cardiogenesis. And these are my 3 specific aims. I think I would leave it here. I don't want to, maybe this is what is the point of discussion in my chalk talk. And basically, I think it's I want to investigate the mechanism of the internal translation initiation, where we put these Yamanaka factor and want to see what role it plays, how it regulates these 5' UTR to produce iPSCs, and how that fragment, when you put some stressors, how would affect cardiotoxicity. So the second aim would demonstrate the role of these isoform in facilitating the membrane electrogenic maturation, because connexin is also involved in trafficking of some of the ion channels to the membrane and also gap junctions. And also the third aim I would want to study in more detail about how doxorubicin can differentially regulate these 20 kilodalton isoform that may be potentially associated with cardiotoxicity at a very low non-toxic dosage. And so the question is, how does Yamanaka factor dependent connexin 43 regulation synchronize reprogramming, and how does this catenane signaling regulate connexin 43 expression during differentiation? So far, I believe there is a yin-yang of 20 kilodalton. Isoform means like there is sometimes it goes up, and it would affect, how would it affect the reprogramming, or if it's going down, or how that would affect. So it will help us understand the mechanistic part of how gap junction or connexin 43 can regulate reprogramming to a better, to further our understanding of its role in pluripotency. So my larger vision is based on these results. I also was very curious about whether these alternate translation is a general phenotype for other ion channels or not. And when I researched, I found that there are internal methionines even in these channels in their 4th transmembrane domain, not 4th, the last transmembrane domain, which also has a C-terminal fragment. And the C-terminal fragment of all these ion channels play a critical role in not only in regulating these ion channel activity, but also they play a role in cell growth and proliferation. But that part of mechanism is not at all addressed in research, so it's an open field. No one, I didn't find any so far, any research associated with these internal translation sites. So my research vision now, towards the end, I would like to elaborate is how the cell regulate these membrane channels to maintain their stemness in development and disease. That's where I see myself in for future. So the significance of this research would be that we will be able to identify the mechanism of these alternate translation initiation, which would be critical for stem cell development of our understanding of reprogramming. And then we can have a pharmacophores that can target these 20 kilodalton fragment isoform to scrutinize their role in cardiotoxicity, which could be transformative in clinical impact on cardioprotection. And then the understanding of these alternate translation initiation of connexin could be applied to other channels and their role in reprogramming and cell fate determination, because ion channels are expressed on the membrane, and they're the first gatekeepers of how the cells respond to the external milieu of a cell. Overall, these emerging mechanism of N-terminal truncated isoform could be generating a protein diversity from single transcript may have implication in health and disease. So I think with that, I'd like to acknowledge some of the people who are associated with this work and in the past have been instrumental for me to reach this stage of presenting my work to you. So I'd like to thank my lab team member, Stacy Edward, who was very well-trained in doing iPSC work and cardiomyocyte differentiation. And she was very instrumental in that MEA paper where I showed you the recording of action potentials. And Viviana Zoklover, who is the postdoc in my lab, was the one also very well-trained optical mapping electrophysiologist. She was with Halife's group, and I happened to meet her because she was in the city, and it's like, okay, do you want to work on this project? Interesting. And she was like, yes, let's do it. And then she was there for the whole time for getting that paper done. And she's moved on for right now. She took another position. And Sean Ryan was a technician in our lab, and John Cook, who was the program analyst. He didn't have any training in analyzing any of that data, but he was just an analytic team who was available to me. So I trained him how to analyze this data. So he helped bring this program. So we now have I didn't mention during my talk, but we have a software, MATLAB software, to extract this data and analyze this data. And also Don Conrad, who initially was very excited to patent this, but then it fell through. I hear from a lot of people asking me, have you patented that? And I said, no, now it's too late. So the funding was provided by some Aurora Foundation internal grant and Cardio Oncology Research Award. These are all internal funding we were provided. And recently we got a travel award from Multielectrode System, the MEA, who provided us a travel award to present the work at AHA. And based on this work, we recently got a funding from FDA to screen some cardiac or potential drugs, small molecules effect on the electrophysiology of these. And we have some collaborators in the math department to help us analyze the data. And patch clamping work was done at UW-Madison by Ravi Vidyanathan. He was my colleague when I was doing PhD. We did PhD together, and he's like, yes, I will want to patch your cells on the one that you're getting action potential or anemia. He helped us do that. And I finally would like to thank my mentors at Hopkins, especially David Yu. I think you forgot to mention, but he was, that's okay. He was instrumental in my training, and he was one of the great mentor I had during my postdoctoral years. I did postdoc with also Leslie Tang, but I spent a lot of time with him, and I think we all know that he's no more. But he was a great mentor. He was a calcium channel king, I would say. And then finally, I would also want to thank Gordon Tomaselli for providing me the opportunity to learn about IPS technology and take it to further do my research. So, and also I'd like to thank you all now. And I would say, let's go Hokies. And thank you. Any connection?
What's your thinking about like the role of poetry, reprogramming versus differentiation? So you said that it can speed up the reprogramming with the Yamanaka factor, but also it can maybe participate to the differentiation into cardiomyocyte. So do you think it's a matter of where connective poetry is in the cell, or maybe connective poetry versus 20K, and that would balance going more into reprogramming or differentiation? What's your
I think I would say that at this point, the translational aspect through phi prime UTR would play a big role, balancing those full length and 20 kilodalton fragment production. Say for fibroblast, like or somatic cells where you begin with, they have much less amount of connexin or even gap junction channel on the membrane. But when you put these Yamanaka factors, that reverses. So now they start producing more connexin and more gap junction. But no one has looked at their phi prime UTR or translational level, like how are that appears. You know, we don't know whether the short or long or medium form of the UTR, which form is existing, and whether it's inhibited or activated. I would say that I would first initially see whether how is that changing, but definitely that would also influence where connexins are in the reprogrammed cells. But through the literature, we already know they are on the membrane. But when they form gap junctions, they do communicate with each other and maintain these pluripotency. But when people have inhibited those gap junction, they don't lose their reprogramming capacity. So it's hypothesized that the reprogramming could be through the gap junction independent function of connexin 43. And that could be in the reprogramming status. But in the differentiation, it's well known that as the IPS move towards the cardiac lineage, as other ion channels are expressed, connexin 43s are also expressed to maintain the communication from cell to cell and their electrical. So did I answer your question? Okay.
Yeah, actually, a really nice question. So, when you're measuring local fields in a neural setting,
Neural?
yeah, from like nervous tissue, they're composed of like low frequency and high frequency components, where the high frequency component is more associated with action potentials, not necessarily generally. And then there's a low frequency, which is more like dendritic potentials. But so from your field potentials, it looked like it was mostly composed of a high frequency component with like spikes. So I was wondering what that waveform would look like if you zoomed way in on it. Like, does it look like a spike waveform, or?
Oh, that's very, I think, neuronal question. Cardiomyocytes are not as, I don't know, if you've looked into the neuronal action potential, they are happening at much higher, their time scale is much smaller, so they're kind of, so for,
What are the frequency components of a cardiomyocyte?
maybe, I'm not able to understand your question, is the frequency,
Like, what does it look like if you filter it at below 500 hertz or above 500 hertz?
So if you
Oh.
did a power spectrum, where would most of the power be? Right.
So I didn't see a difference as much in, we did not analyze, to answer your question. We don't know, especially with this technology, but I'm sure people have done that in patch clamp and in optical mapping. I think it shouldn't make a difference, but I don't know at this point.
Okay, great. Well, thank you all for your time, and let's thank Joshi.
Thank you.